So good morning, ladies and gentlemen, and thank you to One Nucleus for inviting me here to speak. I do understand that there's probably about 60% of the audience who are from a very, very scientific-based discipline. And I was just actually thinking that my talk isn't really highly scientific, but then thinking about it, I think regulatory in itself is a science, or possibly even an art. And I think it's a great thing that everybody is in the labs working away, really trying to discover some of these new therapies that really are needed, particularly in some of the disease areas such as oncology, and really to bring these medicines through. But I think it's something that really people have to recognize that science is one thing, but then you have to really bring it back into that regulated environment in order to really make sure that the product that you're looking at early stage development can come through as a true therapeutic option. And John's already mentioned that in all of these years, there's actually relatively few that do get that far along the development pathway. So why is that? And maybe I can answer a few of those questions as I go through my presentation. As I was getting the slides together, I thought, right, okay, regulatory, is it of interest to many people in the audience? Maybe not, I don't know. So let's try and think of some interesting things to say to start the discussion off. And I did find a few quotes as I was doing a little bit of Googling around. They may not be up to date, but I think they have some true themes there that are relevant. So MAbs are the hottest segment of the biotech industry. And I think you'll see various figures quoted, but in this particular one, it was revenues in excess of $25 billion. And over 150 MAbs being developed in over 200 indications. So really, there is still the effort there to get these products to the market. But development times have increased from a mean average of 6.7 years to 8.3 years latterly. And now we're seeing possibly a new generation of compounds coming through, and I say new, but these are the biosimilar MAbs, and they are really being seen and developing their position for substantial growth. There are a lot of the blockbusters coming off patent over the next decade, and these compounds the biosimilars do have that potential to reduce healthcare costs, so there's a lot of interest in the biosimilar arena. So on that background, and with the knowledge that monoclonals are not new, are there really any challenges that you have to face in 2013? And that was the question I had to actually ask myself after a long history in regulatory affairs. What really are the challenges now? Because they have been around for many, many years. It's a long time ago that I think there was the mention of the magic bullet. I mean, we're so advanced now. And as you've heard already in the initial presentations, we've gone from the murine-based compounds right through now to fully human monoclonal antibodies. So tremendous development. We've got extensive experience now in the manufacture and testing approaches. And David gave his great presentation from MedImmune, how they're now trying to actually control some of that variability. But we have got extensive experience in manufacture. There are many, many guidance notes that regulatory people can read and that have to be adhered to a certain degree around monoclonal antibodies and biotech products in general. So on that basis, are there really any regulatory challenges? Well, I think, yes, there are. As we've already heard, that monoclonals are very, very complex structures. And the key thing in the mind of the regulator is making sure at the end of the day that those complex molecules, once they get into the clinic and to the market, really will have that evidence of safety and efficacy behind them. But I think even from some of the presentations this morning, we're seeing that really still it's not fully understood regarding the specificity of these antibody products, how they are functioning, their structure, some of the changes that are happening during storage or manufacture, and how that's really going to have an impact on their mode of action, their efficacy, and their safety. We know that the development times are long and the programs are extremely costly. So companies are naturally looking at ways that they can accelerate their development programs and development pathways. And to do that, I really think they need to work very, very closely hand in hand with the regulators. And the regulators' challenge is really looking at trying to find a balance between the requirements they have, the confidence that they can have in the safety and efficacy, but recognizing that there really is still an unmet medical need in certain areas. And I think one of the interesting challenges, certainly in the company that I'm working, where we see a lot of products coming through, there seems to be this increase in looking at rare diseases, rare conditions, and they in themselves bring tremendous challenges. As a good regulatory person, I like to structure things, and I think we're brought up in a regulatory world to think about quality, non-clinical, and clinical aspects. And that's how I've actually structured the next set of slides. But when you're talking about monoclonals or biologics or biotech products, you can't actually think about these as discrete units. Everything has to be taken as a continuum, and also continually backtracking. So even when you're in the clinic, you need to backtrack to the quality, you need to backtrack to your non-clinical, and really make sure that everything you're doing along the way is still fulfilling its needs. But if we do start to look a little bit at some of these specific aspects, and just taking the quality challenges to start with, as I say, there are many, many guidance notes out there that you can follow on characterization, production, specifications of monoclonal antibodies. But now I think as we move forward, and during the course of today, you're probably going to hear about some other more advanced monoclonals like the fragment products. And with all of these advances, the regulators really are now expecting that you're looking to use advanced technologies to really try and characterize these products so that they can really get more of a comfort level as to how they are going to act, whether it's through production and some of the changes that could potentially happen, or when they do get into the clinic. I'm sure many of you, if not all of you, have heard the phrase, the process is the product. This isn't like a small molecule. Any little change in a complex manufacturing process can potentially impact on the safety and efficacy of the product. Certainly, I think it's going back now about 8 or 9 years ago, which is quite some time ago when I worked at Amgen. You know, some changes that were made to the manufacturing process of erythropoietin resulted in some very severe adverse events. So, you know, even with something that is potentially a well-established biologic, you can still get potential changes that have an impact on safety. So the emphasis really from the manufacturing and control point of view is to really look at controlling the processes as well as you can, having some tight controls there, really, really limiting those fluctuations, and hence really gaining a good control on your efficacy and safety. And I did find David's presentation particularly interesting with my regulatory hat on, just really trying to control that at a very, very early stage so that you do get the confidence going through to the clinic that you're not going to end up with a large number of changes. But I think it is inevitable that you will have some changes as you go through development. So what do you do? Because you know, you're working along your development pathway, you're going to go into the animal setting, you're going to look at tox testing, you're going to go into the clinic. But what if you do change your process along that development pathway? Does the quality then really still support what you've done in the non-clinical and clinical setting? And you may find that you have to go and backtrack and do some comparability exercises, certainly on a quality level or maybe even on a non-clinical level. Really, really looking to see whether any of those changes would have a potential impact on the safety and efficacy of your product. Another challenge, as we've heard through some of these complex processes, is to really have a good knowledge and control of your process and product-related impurities, because it is certainly some of these impurities that may have the potential for adverse events, such as infusion site reactions or enhanced immunogenicity, which in itself is a complex issue. And I think certainly for monoclonals, what we're finding is that even going into early clinical studies, the demands of the regulators on the CMC side is getting higher. They really do need to have that comfort that you can go into the clinic. If we look now at the non-clinical challenges, I think everybody sort of felt fairly comfortable that, you know, maybe it wasn't that easy to find a relevant species. Okay, let's sort of default with the human monoclonal antibodies to the primate as your acceptable model for non-clinical testing. And I think all of that was sort of thrown in the water with the Northwick Park Tigenero episode. So again, from a regulatory point of view, and the regulators really looking for you to come to them and really have a knowledge of why you've used whatever species you have chosen to use in non-clinical testing. I mean, obviously you need a species that is going to show some sort of pharmacodynamic effect, and hopefully, and I say hopefully in this setting, you will start to see some toxicity. I think the hardest thing for a company and a regulator is if you don't see toxicity, then what do you do? And again, I think for monoclonals, they obviously need to be very specific to the target, but again, a regulator's going to want to know if there's any potential off-target effects. And for a monoclonal, you've actually got two, if not more, aspects that you really need to look at in the animal setting, and that's really what's happening with the binding via the Fab part. Is it binding to the actual receptor that you're aiming at, but also the FC receptor and what's happening in that domain. And again, probably not actually solely relevant to monoclonals. I think you see this in every development that you're undertaking, no matter what the molecule is. And that is the difficulty of defining the dose to go into your first in man in the clinic and defining an adequate safety margin. But it's enhanced with monoclonals because of the difficulty of really finding that species that is relevant for non-clinical safety testing. And as well, monoclonals can have a very unusual dose-response relationship. And sometimes you do find that at a lower dose, you're getting a pharmacodynamic effect that you maybe would not have anticipated. So really, again, from a regulatory point of view, it's understanding the kinetics, the pharmacodynamics in the animal setting before moving forward into the clinic. So that then brings you on to the clinical challenges on that background, keeping the quality aspects in mind, potential changes in the manufacturing process, etc., the difficulties of non-clinical testing for a monoclonal antibody. So how do you really then move safely into your first-in-man studies? And again, as I say, with before the Northwick Park episode, I think everybody was sort of getting into that comfort zone. And now we do actually have the heightened vigilance around going into first-in-man, particularly with monoclonal antibodies. And there is a guidance out there with regard to trying to really identify the potential risks of your product before putting it into that setting. Really looking at the mode of action and identifying if there is the potential for multiple signaling pathways so that you won't get that type of cytokine reaction. Looking across, as I say, the quality and trying to control now at a very, very early stage the potency so that you don't get untoward fluctuations in potency, that the batch that you are using from a clinical point of view is actually reflective of the batches that you've used in your non-clinical testing. The non-clinical really looking to see that you have, to all intents and purposes, done as much as you can to find a relevant species, or if there isn't a relevant species, is there a different model that you can use? And then looking at your proposed population and how you are actually going to manage going into the clinic. And I think now it's not uncommon to see a very, very cautious staged approach, particularly when you're looking at dose levels. I think another challenge that is starting to come to light is, I've called it personalized medicines, but really looking at some of the markers for clinical efficacy outcomes. And certainly there seems to be this drive from the regulatory point of view to really see if there are potential markers that will identify a population where your product will be particularly effective. And I think there are some examples out there. The one I put here is the Amgen product, where the indication is specifically for the non-mutated CRAS in colorectal cancer, so a very, very, very defined subpopulation. And as I've mentioned, the rare diseases, which I think from a clinical point of view, you could probably spend a whole day talking about those challenges. The other area that I think needs to be uppermost in people's minds as they do go through their development process is really some of the pharmacovigilance aspects, which I think tend to be pushed to the side until you get very much later on in your development. But I think if you start from the outset really thinking about some of these potential safety concerns and really looking at managing your risks over the whole life cycle of your product, looking for potential immunogenic reactions and how you can monitor and potentially manage those. And certainly with the introduction of some of the biosimilars now, what is becoming quite important is a clear identification and traceability of your product so that you can ascertain what product has caused any potential adverse effect. In the regulated world, there is now a very, very strong emphasis on a risk management plan. It's a crucial part of your regulatory submission. And it's not a static document that you put in with your submission and then you forget about it. It really is an interactive document. It's a progressive document that lives with your product. So you start to look at some of the potential risks. You look at how you can manage those risks and you monitor those risks when you get out into the commercial world. And I think you couldn't talk about clinical challenges without not mentioning immunogenicity, which again, people may say, well, that's an old hat, everybody knows about it. And certainly with the human monoclonals, as was mentioned earlier, the whole concept is that you are potentially reducing the risk of immunogenicity when the product goes into the clinical setting. But immunogenicity in itself is a complex issue. So yes, I think with most antibodies, human antibodies, you are seeing that reduction in immunogenicity, but there are still immunogenic reactions occurring. So that really is sort of looking across the development aspects, but I thought it would be quite interesting as well to look at some of the relatively new challenges, as I've mentioned earlier, in the context of the biosimilar monoclonal antibodies. And certainly when I was working at Amgen and some of the legislation was being looked at for the introduction of the monoclonals, and we used to sit there saying, oh, there'll never be a biosimilar EPO. Well, there are several now that are on the market. And I'm not going to pretend that these diagrams here are actually the ones that I've indicated below them. I took them off the internet. But it's really just to illustrate the point that, I think people really thought that, yes, filgrastim is a very, very simple molecule. Definitely, no problem, there will be a biosimilar. As I say, EPO, really people were saying, no, never. It's a more complex molecule, processes the product. How can a biosimilar developer actually try and copy or produce something that's similar to something so complex, particularly in the light of the pure red cell aplasia that did occur. But oh no, we have them on the market now. So my question is, will there ever be a biosimilar monoclonal antibody? Will there? at the end of the day, you know, the developers, the originators know their processes so well. We know these are extra large complex molecules with extra complex processes. As I say, the originator knows that process inside out. They know what specifications they're setting. So how can a biosimilar developer actually produce something that is very similar? Well, everybody is watching to see if this is going to happen. In July of 2012, a Korean company did get authorization of its anti-TNF alpha monoclonal antibody in Korea, and in March of 2012, it was submitted to the European Medicines Agency. I don't think anything has appeared yet, but certainly I would have thought come March of 2013, we should be hearing some rumblings as to whether that has been successful or not. There are so many companies looking in the monoclonal antibody biosimilar space that if this one is successful, then certainly yes. You're going to see a lot of them coming through to the market. And it's interesting just looking at some of the biotech products that are coming off expiry over the next few years. And a fair number of those, as you'll note, are monoclonal antibodies. So with all of these potential challenges in the background, I think that probably answers a little bit of John's question as to why more have not gotten to the market. But I think from a regulatory point of view, for me, it's actually looking at those challenges and seeing how you can mitigate the risks of those challenges. And I think from the regulatory point of view, some of the messages, or certainly one of the key messages that I think should go across, is really to look at your regulatory strategy from the outset, to see how that regulatory strategy will map into your development program, and how you can actually then look at potentially moving quickly to the market. But on the back of a really robust development plan and looking at the end in sight, looking at where you're aiming with your target product profile, what is your indication, what is your potential dosing, what is an acceptable level of adverse events, and really mapping your development plan from the outset through to marketing. A message that I always do try to put across is engage with the regulatory authorities early. I think people are getting over those barriers now that the regulators are there to prevent them doing things, and you know, we shouldn't go and talk to the regulators because we might hear things that we don't want to hear. But from, I think, the regulators' point of view as well, the earlier they see these compounds, the more familiar they do become with them, the easier the ride you're going to have. And I put down here, know where your data is. Certainly not for the MedImmunes of the world, but for some of the smaller companies. You know, we see companies that really don't even know where the reports are, and, you know, has the report actually been written up? Well, if you're in that position, that can severely delay your development programs. So some of the future regulatory challenges. Well, I think people now are looking really much at platform technologies and how they can actually look at almost a regulatory authorization of a platform and then building on the fragments to that. I think something that really people should be looking at are the alternative approaches to non-clinical testing. What if you can't find that relevant species? What do you do? Is there a transgenic model? Is there a disease model that you can use? And as I say, the move is really towards, in the clinic, looking at validated biomarkers to really target to the relevant patient populations. So are there any regulatory challenges in 2013? Well, you can draw your own conclusions, I think. I think there are. I think there are still challenges in the quality non-clinical and clinical setting, despite the wealth of knowledge that we do have, because technologies are changing. There is a heightened awareness and focus, certainly in the regulated world, on post-authorization vigilance so that therapies can come to the market earlier with the vigilance happening after authorization. Really look at robust development plans and use those as interactive plans as you work your way through your development pathway and map on your regulatory strategy to get the best of both worlds. And really do open up that dialogue with the regulators. Thank you.